Hi, good morning to you all. Just hold on for maybe 5 minutes more. I'll be back. Yeah, I'll just
Good
finish.
morning, sir.
Okay, sir.
Good morning, sir. Yes, sir.
So, good morning. We have been up to now studying this is by prospecting class, and you remember we started with saying that we will be manufacturing and making a goods which will be sellable and which will be beneficial to the society and to our economical growth. In that process, I think up to now we have been last 6, 7 class or 10 class, we have been concentrating on microorganisms. So today we will take a break. I think what we did is we started from just beginning of collection of soil to the product isolation, that is upstream, downstream, fermentation, construction, and all that we finished. The next thing which we need to do is now, I think we will come back to this because you should not forget that's most important thing we started with. We will come back after 2 or 3 classes, and we will also touch upon again a fermentation of antibiotic. This is one of the topic, and which is again very favorite in an examination question point of view. So, but now I think before we really see, everybody need not be just interested in microorganism related, correct? So today another big resource is the plants and animals. I think animals are much lesser as compared to plants. Animals, then it comes under farm breeding and those. That's a different aspect altogether, which is not a part of our bioprospecting. And right now, what we are our the framework which we are working in this. So major thing will be a plant. So anybody has any questions around previous one or this one? So I think first I'll touch upon that because today's class is not going to be a big thing. This plant related thing, I will today introduce you all, and then you all have certain, I think, what do you call assignments? Assignments which there is a marks for assignments and presentation, correct? And the class test. These are the three factors which are internal marks. So I think we are going to do more of a pharmacognosy, which is a word which I'm going to introduce you. I'll introduce you to the field today. And the part of that you all will be doing it in your assignments, and then we will see how much detail we should go into it, correct? So that is how I have planned. Any questions or anything which you have not understood? Can you all hear me?
Yes, sir.
Yes, sir.
Yes, sir.
Okay. So nobody has any questions, correct? So I think I'll start that the new topic which is called as pharmacognosy, which is of the plant. So yeah, I think somebody wanted to talk something.
Sir, what is spray drying in fermentation?
Huh?
Spray drying.
Pre?
Sir, spray drying.
Spray drying. Okay, good. I think you all have received the notes which I have sent.
Yes, sir.
Yes.
The Word file which I have sent. I think one of you requested that though I gave you all the charts and the notes, but I think Word file which I sent, did I send it? You all didn't check. See, if you don't check now, and then next just 2 days before exam, you all will call me up saying that sir, वो notes आपने भेजे थे मिले नहीं हैं अब कहाँ है? तो I will not be able to send you again. For me, it is impossible for to.
Yes, sir, it's there.
हाँ
It's there.
मिला थे नोट्स मिले आप लोगों को?
Yes, sir.
हाँ जो मैंने एक word by word, and I think this spray drying came up, so I think it's a good thing. I think some of you have read it, so I'm very happy. So I'll explain this Tanvi's question. Is that Tanvi correct? Tanvi Sharma.
Yes, sir.
Tanvi, so overall I think this class. So you will please read this and keep on asking question anytime in any class. I will be able to answer. Spray drying. So what we do in certain products which we sell it as a crude. I'll explain you with the yeast which you get it in your house. Correct. So you remember in a fermentation we talked about upstream, and then we talked about another aspect where the cell mass is important or broth. Certain cases, like let's put it as a yeast, yeah, which you are getting in your home in a packet, correct? Now, when you complete the fermentation, in that case, your whole concentration will be in getting the yeast, number of yeast cell, large number of yeast cell if you get, the higher the biomass you get, the higher your yield will be, the more more packets you will be able to fill up, correct, of yeast and sell it. So, so in that sense, what we do is once the biomass is ready, we first wash it with water. And this biomass is yeast, correct? Because you have grown yeast because we want to sell yeast. And in this case, we do a spray drying. So there are multiple ways of spray drying can happen, but I'll explain you one of the most simplest way. you heat up the drum, that there is a rotating drum which is at the temperature of 40 to 45 degrees centigrade, and it can be kept in a vacuum also. And then with a sharp nozzle, you spray this cell, the cell mass which you have got, you dissolve it in a small quantity, and you know that 10 raised to 9 cells per 1 ml. So similar, you dissolve it in say 10 liters, and now you start spraying into this area where the drum, warm drum is rotating. The area is with a vacuum. Yeah. And when you spray it, what will happen? See, the vacuum and the low temperature both will help in evaporating the water, and you will get the powder. And this is what the spray drying, one of the way of spray drying. Yes, Tanvi, did you understand?
Yes, sir. Thank you, sir.
Yeah. Anybody else? Yeah, so this is related to again downstream processing. Yeah, even it can be done with the broth, but the broth being in hundreds or thousands of liter, spray drying is very rarely done. Right now, when I'm talking to you all, I can't recollect any of the example which I can give you where the before processing a partially processed proteins, once the suppose if our interest of product is of interest, and if it is partially purified using the column chromatography or gel filtration, the last step in the before marketing, I think you have to, normally you don't market in a wet form unless it's an alcohol or a beer which you bottle it and sell. But many of the fermentation products are sold even in injectables of the enzyme. You sell it in the form of dry powder. Yeah, amylase, for example, we have been continuously. So they all will be done in a, either they will in a final step, they will be crystallized or they will lyophilized. Lyophilization is again at a low temperature under the vacuum removal of water. That's a lyophilization or crystallization, which I have explained last time, and this spray drying. So these are the some of the method, final step method before you make your product ready for marketing. Now you all go through again, the some of you may not have got time to go through all those notes, but please go through it. See what I'm sending you, there is no standard book, or there I can give you the list of multiple books. I don't think none of you are going to have a time to read through it. Otherwise, I can give you Kasida. I'll send you the list of books unless if somebody is really depth interested in it. Once the college starts, all those books are there in the library. So you can refer to the books, but please first read these basics notes, yeah, and just try to correlate with our discussion in the class. Okay. So I take it for granted for right now. Today's class is not going to be very big. So again, if somebody has a question on a previous thing, it's good to go ahead, because today we are literally changing the gears from a microorganism to now we are coming to a macroorganism, and the first thing we are taking is a plant. Though I'm not going to take animal, because animals are not really, as I explained you, not much use for unless you are in Northeast. Yes, I have submitted one. I have approved one project when I was in DBT, Department of Biotechnology, for insects, which they used to increase the protein, because in Northeast they eat those insects for a protein. It's a big source of protein. Like हम लोग जैसे मूंगफली खाते हैं ना. I think Athibo might be there. He might be knowing. So in Manipur University we gave that project. So that insect protein is very important. So that was some. But I'm not going into today. I don't think in this class we are going to much. I'm going to teach anything on insect. But yes, plant you all know. We have been using right from something, all the plants we are using, the whole Ayurvedic, homeopathy, a lot of things depend on our plant medicine. Plant-based medicine is a very widely accepted, and if you see in a Chinese and Indian Ayurveda, it's all based on plants. So we'll be doing that today. Any questions? Okay, then what I'll do is I'll say I think today you all will read the slides. Okay. Devanshi, you first read few slides, then Tanvi, and then Nehal. Okay, agreed?
Okay, sir.
Okay, fine, good, thank you. I'll put the slides on. Wait, huh? I'll share the slides. It's very basics, I think. and very simple. The first lecture is just an introducing you all to the pharmacognosy, the word, which is 2 mark definition. Take it for granted. I'm leaking the paper today. So, can you read the word? Can you see the slide? Pharmacognosy.
Yes, sir.
Good. So then I'll switch to the first slide. The pharmacognosy is the word. I think first we'll try to learn this word and take it for granted. Get 2 mark or definition, or if it is a 6 mark or 8 mark question on pharmacognosy, as soon as you put a definition in your first paragraph, I think you get straight away 2 marks for that. So now, yeah, please read it.
Pharmacognosy is derived from two Greek words, which is pharmakon and gnosis. Pharmakon means drug, gnosis means knowledge. In brief, pharmacognosy means knowledge of drugs. It is a study of drugs that originate in the plant and animal kingdoms. Pharmacognosy is a branch of pharmacy which deals with the basic resources of medicines from nature, which is plants and animals, and their uses as medicaments from the ancient time to present day.
Okay. So I think this is very self-explanatory first slide, but I think it sets the stage very well. Correct? And it's a Greek word. So pharmacognosy. The pharmakon is knowledge. I mean, yeah, pharmakon is a drug. We all know pharmakon is a drug, correct? We, as soon as somebody say pharmacy, pharmakon, we know it's talking about drug. And gnosis is knowledge. So I think it's a very, very self-explanatory, correct? So I'll shift to the second slide.
Pharmacognosy may be defined as an important branch of pharmacy which deals with the study of structural, physical, chemical, biochemical, and sensory characters of natural drugs of plants. It also includes a study of their history, distribution, cultivation, collection, identification, preparation, evaluation, preservation, use, and commerce. At present, pharmacognosy involves not only the crude drugs but also their natural derivatives, that is, pure compounds or constituents, digitally isolated glycoside, digitoxin, raw Volpia root, and its purified alkaloid
Reserpine.
yes, and thyroid gland with its extracted hormone thyroxine are all part of the
Is a part of the pharmacognosy, we can say, correct? I think it is getting covered. So, this I need to explain. So first slide, I think it was, first slide was, I think, very self-explanatory. And this slide, I'll go very slowly, because each word is loaded. And here I will try to give you explanation. Now, don't worry, as much as you remember is fine. But I think if you remember this slide and then it will be easy to expand it, correct? Pharmacognosy may be defined as, and first we gave a classical definition. Pharmakon means what? It's a Greek word and all. It may be defined as an important branch. It's a really very important branch of a pharmacy. In India, now I'm not reading on a slide. Yeah, in India we have an Ayush. How many? I hope that some of you have heard about Ayush department. Which deals with an Ayurvedic, homeopathy, Unani medicines, and that's a big department. And there are a lot of funded projects, and as you all will be coming, as you will grow, I think it is good, this is a good time for you to get introduced to Ayush word, because in India all the pharmacological, this pharmacognosy or plant related projects, and plant related especially medicinal projects, are funded by Ayush, and it is a good, it's a ministry of Ayush. Yeah, it's a big ministry, a fairly big ministry. So just to give you this briefing I gave you so that you get interested that how important it is nationally and in Asia point of view, I think even including Japan. A whole overall Asia, I think we are very, very much oriented towards plant, and it is picking up. It was always there and it will remain there for a very long time. So coming back to this slide, so which deals with a study of structural. Now, plant will have what? Roots, leaves, bark, correct? So these are some of the very simple things. But as you all may be aware, because this is every day to day in our life, that every plant will have a very different use, like every plant material, which different structures if we talk about. Even in the leaves, early growing part of the leaf is having a different medicinal value, and that is very tender leaves, or otherwise and very yellowish and which are almost falling down, or completely grown up leaves will have a different kind of. So that level of structural, but right now enough for you to understand, it is important to know that for each medicine, it need not be the whole plant need to be used. It can be leaves, it can be bark, it can be roots, and this is how it is a structural. Physical, exactly correct. As I told you, whether it will be whole leaf we are going to take or a different extraction we are going to take, that will fall under a physico-chemical and a biochemical aspect. Yeah. So this is characteristic of a natural drug plant in animal origin. It also includes study of their history. Now, it is very important. If you see from the, I never told you about when we are doing with microorganism, that history of the microorganism is really important to us, correct? I never told you. But when it comes to plant, I think historical importance is very high. If you see our Charak Samhita, I don't know whether how few of you might be knowing about Charak Samhita. Charak Samhita is one book which has all the information about our plant resources and how they can be used. So the history, even Chinese, if you see, if you really see the plant really come, if you look at the Ayurveda drugs, it comes more from this region, Asia region, and Asia has a better historical understanding and knowledge, and really literacy was much higher in Asia as compared to the Western world. And that is why the historical wisdom is very important when it comes to this plant. I will give you one example if you all are interested, where I worked on a dengue plant, but that I'll come little later. Let me not disturb this basic understanding. Yeah, the history of the plant, the distribution, very important. A ginger, if you take it from a northeast, I don't know whether I told in this class or not, but I was really taken up by that. Like, if you take a ginger or a citrus fruit from a northeast versus in a south down Kerala, I think both the gingers have a completely different chemical structure, physical nature, and multiple. One of the ginger is more used in a pharma industry, and another ginger is more important in a household. Like whether a pulp is important or whether the juice is important. I think I've given in this class. I don't remember whom I've given, but this is a very classical example which comes to my mind when the geographical distribution comes into the picture. And I think it's self-explanatory. I think some of you, if you are with me and thinking, see, if the flora and fauna definitely affect the plant in which soil you are growing. Ratnagiri Hafus is very different as compared to our Chosa, which comes from a North India, or our Daseri is very different from any of the South mangoes. I'm just taking example of mango. So you all know, or that is how you know that, like, soil, the atmosphere, they have a big impact on the plant. And that is why it is very important to know, even more important to know firstly about the history and the distribution of plant and where you will grow. And once you know that cultivation and collection is associated point, which I am, when I elaborately discuss you, I'm sure you must have understood that you have to cultivate. If once you identified that this plant is of interest to me, the cultivation and collection of this plant is very important. From which region you will collect, whether you will collect the leaves, whether you will collect the roots. So all those collection will come into picture. Identification, oh God, this is the most difficult. The whole patent of ours got completely invalidated at one stage when another person came in and said that this identification, because botanical identification even today is in very doldrums. So identification is very, very important. Preparation, evaluation, and preservation. These are preparation of the final product which you will supply it in the form of what form. We'll go into detail of what preparation, how we'll prepare the plant. And evaluation, correct? Evaluation is again goes back to our screening model. Screening model is a fundamental, right? Correct. If you are looking for an antiviral compound or amylase from the plant, I think you need to use the screening model. So I think we spend a lot of time on screening models. So if you are even looking for, here the animal model sometimes come like fever. Yeah, fever, because in Charak Samhita most of the things are being reported as a jwar. So what you do is ultimately you have to really either you look as an antiviral compound because virus causes fever, or in animal you create and increase the temperature using chemical methods and then give this plant extract to see whether the fever is getting reduced. Like when you take a crocin, yeah, paracetamol, when you take, how your fever comes down. So whether there are many plant material which are more effective than this crocin, and I myself have seen. That's why I'm telling you. When we were in Northeast, 4 years I've been going to Manipur, and I was amazed to see their knowledge on plant. Okay, so then you need to preserve, correct? Because we are talking about commerce. We are going to sell it, so we have to prepare. The preparation should be acceptable. Then we have to evaluate and label it, and then preservation we has to be done, and we have to sell it. Now, I want you all to ask some questions because this was very elaborate, a small, it's like a whole Charak Samhita, which is book which we can't lift that heavy, heaviest book you have seen probably in your life. It's that big a book. So that I've tried to put it into one paragraph and try to explain you. Yeah, and the down which she read about the presentation involves the not only crude drugs but also natural. That will come to later. But from this itself, if you remember this, I hope that you all must have understood the importance of having a very concise and like lyophilized or dried powder I'm giving you, but it has a lot of material. Every word has importance, and I try to explain you structural, physical, chemical, biochemical, and then history, distribution, and that's why the different colors were put. Please ask some questions.
So what exactly do they mean by sensory characters of the drugs?
Sensory characters of natural drugs of the plant. Yeah. So
Yeah.
I'll explain you. Let me give some very easy example. I don't want you to give a very complicated. Okay. One thing can be stability. At least that if you remember. Though the plants normally what we get when we talk about a crude plant and their extraction, that is going to be a very stable, but as you purify it, it becomes very unstable. So that is one part of it. But there are more complicated things associated with the sensory character of the plant. It's not a natural drug when they say actually the plant and the plant material. Now what plant come in front of you is like neem trees, and when I talk about medicinal plant, I'm sure if you are thinking, closing your eyes and thinking, but there are certain very delicate species like almost like a mushroom, and they call it into the plant, and in that there is a sensory characters which also slightly related. When he is using here sensory characters, I think he also means that animal related origin, and that's why that you noticed very well that I was remaining silent on sensory because I don't want to touch upon animal origin related drugs, correct, right now. So it is more here in this paragraph. You can remain silent on sensory and animal because I don't want to touch today much on the animal related, animal origin drugs. Does that answer your thing?
Yes, sir, answers.
Yeah.
Thank you.
Yeah, I didn't try to explain you. In fact, I told you how to remain, why I'm remaining silent on sensory. Yeah.
So,
Yes.
sir, what is identification process like? Is it about how we are identifying the plant?
Correct. Plant identification, botany. If you learned little bit botany, I think I'm sure it has been taught to you. The identification of the plant is a very complicated process and a very, very mixed up process. Now, if you ask me in bacteria, is it that difficult? No, because we have a very good Burgess manual and very nicely written down system from the Western world. And also we have, a nicely organized now in a modern world, a DNA analysis, RNA fingerprinting, or, ribosome related identification. So detailed identification is possible. When the plant, if you are keeping an eye, or I'll share you with you a news item which was just 2 days back, was there that new plant species were discovered in India by Punjab University, and plant was from, what you call, a goddess, mermaid, goddess of sea or something. So they named it after that. By telling you this, what I wanted you to tell is it's not completely developed process today. So you all have, of course, you must have learned a botany, correct? Identify right now. This is beyond the scope of to teach you classification or identification of a plant, but it's a completely branch of botany where the identification systems are developed, like, animal kingdom, plant kingdom. You have learned. I
Yes,
think
sir,
you all
we
have
have
learned,
learned.
correct? Huh? So it's
Yes,
it's
yes.
that. So that's identification. And as I was telling you, I was just trying to share my experience, and that was little. Again, I will say repeat if you missed during the when I was reading. That, I was working on that through Ranbaxy, we were working on a dengue plant, and we identified as a season chlorosis. One of our botany group has identified, and we filed a patent, and we were working through Ayush funding onto that project for dengue. And recently, after I left the whole job and recently, 3 years back, I received an note saying that that identification was wrong, and it was re-identified as this. And I used to go to Manipur, and so I'm in touch with multiple people in the identification of Ayush department, medicinal plant identification. So it's an upcoming process. Field, so that's a field where you all can enter, and really, if you are entrepreneur, find out a new species or an same species, properly identify and get the patent, or at least that's a good job opportunity. So identification question I elaborately answer. Any other? This question helps me in telling you some kind of stories which maybe will remain with you. बाकी तो ये सब आप लोग पढ़ के बहुत अच्छा लिख लेते हो. मेरा अभी मुझे पता experience है. I've been checking the paper, so you are a master in reading out and putting it into your answer sheets. A digitalized leaf, if you see, come down into the second portion of that, in that digitalized leaf, the digitalized identification. So they take the photographs in a multiple direction, and then now they have tried to computer AI people have entered into this field, and they are trying to digitalize everything. The moment if you are in a jungle and you just take you see, many a times we take a photograph and we put it onto Google, it tells you the name of that plant. Try that. Yeah.
Yes, sir.
Yeah. Okay. Any other further questions? This one slide is power packed slide. That's why I'm spending so much time. If you see the second part, it is talking about the digitalized leaf and its isolated glycoside. So glycoside, risarpin is an alkaloid. See what are the things which comes from a plant is alkaloids, glycosides, hormones. These are thyroid related. Thyroxine, we are still getting it from some plants and weeds. So these are some of the important example also it came. Even it is telling you in this slide that, it's nowadays we are not only dealing with a crude product, we are dealing with a purified product plus some of the derivatives of that plant related. So pharmacognosy is a big topic as much as any other medicinal, our allopathic medicine equivalent to that. Any other question on this slide? So we'll not spend all 47 slides. I will share with you all, but I think important things I just want to cover here. Okay, I think this is easy slide. I mean, please read it.
Pharmacognosy and modern medicine. Simultaneously, advancement in the field of chemistry, biochemistry, biosynthesis, and pharmacology have developed pharmacognosis. Various active compounds have been isolated from plants, which are used in modern medicines. With the advancement of synthetic organic chemistry, most of the active constituents of plant have been synthesized. More than 100 plants are used in modern medicine in various parts of the world.
I think here the pharmacognosy and modern medicine are try to, it is joining with the previous slide, correct? As I told you, we are not dealing with only crude product with our knowledge of advanced synthetic chemistry. And any question, I think this is a very self-explanatory. Take 2 minutes to read this slide, everyone. And if any question comes to your mind, these are like 6 sentences, please try to read it. Oh, sorry. Can you all see the slides? Sorry, I think I disturbed that, right?
Yes, sir.
Yes, sir. I can.
Shall we go to the next slide if there is no
Sir,
question? Good.
can,
Ask.
can you please state some advancements in the field of chemistry, biochemistry, in the
Synthetic organic chemistry. Okay.
yeah.
I can share with example because okay. ठीक है. This whole class, just to make you make interesting, and this is the real world. That's why I'm telling you. पता नहीं आप लोगों के लिए ज्यादा ओवर इंफॉर्मेशन हो जाए तो उसको फिल्टर करके डब्बे में डाल देना, बट आई विल टेल यू द होल थिंग बिकॉज यू आर इंटरेस्टेड. आई हैव टू आंसर दैट इलॉबरेटली. So what is that, synthetic, once you just imagine, you questioned me on that you were telling that if I collect the plant from a different region, yeah, so if you are collecting a dengue plant from Rajasthan and then you are collecting it from down south Kerala, both will have a very an MP. Last we settled down on to Madhya Pradesh plant. So Sesam pleurosis, if you are collecting, identification itself is difficult. In that process, how can I trust the compound which you are, the preparation which you are giving me as an anti-dengue will be consistently, like, you are not going to sell it only one bottle. Correct? We are going to sell it for years together, multiple batches. अब मैं point पे आ रहा हूँ कि why we need and how we use but first it is important for you to understand that why it is required. Now, if I don't have a good organic chemistry or analytical chemistry tools available and applied during the development of this medicine, what will happen? आज तो आप क्योर हो जाओगे. मैं आपको आज प्लांट दे दूंगा, प्लांट मटेरियल आप ले लोगे और मेरा प्लांट प्रिपरेशन आप बॉटल खरीद लोगे. आज आप क्योर हो जाओगे. कल यदि प्लांट दूसरी जगह से कलेक्ट हुआ और उसमें वो पर्टिकुलर एक्टिव इंग्रेडिएंट नहीं होगा तो आप क्योर नहीं होंगे. इट कैन बी फेटल. So in that scenario, the development, one of the advancement is NMR. And NMR is like you all must have heard of 32 megahertz, but it has gone to more than 560 and 1120, more than that. Like, advancement, every day advancement comes up in NMR. So what we do is we do a fingerprinting. Abhi bahut complicated ho jaye to don't worry the class, but you have to take a fingerprinting of the material which you are selling, and then when you are doing a screening, you should know that if this component number 1, 2, 3, and 4 are present in a 10%, 20%, 40%, and 70% composition, then this plant is the best active in my screening model, in animal model, and in human. Once you have this fingerprinting, then this fingerprinting requires a big advancement of HPLC, analytical tools of an organic chemistry. Yeah. So that's one aspect, very important aspect of quality controlling of plant material using an advanced tools, IR, NMR, multi-megahertz, and HPLC. Yeah. So that's one of the examples. There are multiple examples, but I think all those we will not get it into it. But this is one of the examples. This is real life. Okay.
Yes, sir.
Okay, please read this. The person who just boy who read right now asked the question. Please read it. क्या नाम आपका?
Yes,
Sorry,
sir.
I cannot see
Yuvraj.
your. Huh?
Yuvraj Nigam.
Yuvraj, just read this line.
Yes. Applications of pharmacognosy. Plants always provide novel molecular structure, which played an important role for the molecular design and development of novel potent, less toxic or non-toxic drugs. Example, natural morphine has gone analgesic and narcotic effect, but piperidine, a synthetic drug, originates from morphine basic skeleton, is non-narcotic. Plant and animal products give potential biological effect without any undesirable effects. Plants and animal products can be taken with very ease.
Yeah. Just the whole class, please take a minute to read this and ask some question. When you ask, I hope that this method works. If somebody feels that method is not good enough and waste of time, then I can still change the method. But at present, I feel this is an interesting way to go ahead. So I'm using this method. I hope you know analgesic means painkiller, huh? And morphine is narcotic. Morphine falls under a narcotic drug.
Yes.
different drug के classification है. Shall I switch to another slide? So I think what we did is we defined pharmacognosy and then the important different aspect of the plants. Then now we understood the how modern medicine got connected with it. So we gave it an complete right from an historical to the present perspective and then application we are talking about, correct? So you all have seen non-toxic, a completely novel molecules as compared to what we would have got from organism or a synthetic chemistry. It can be a good base material for a future derivatization, which can be more effective, less toxic. So that's adding an synthetic chemistry, semi-synthetic we call this as, because you start with a plant material and then you are modifying it to the next drug. So it is a semi-synthetic chemistry. Okay, now with everything as a limitation, correct? So it's this four aspect, even if you remember, that will be good enough. I'll share the whole slides set, but I think that this is another very important. After hearing all good of things about it, I think you should learn the limitation. So one of you, please read it. Somebody please volunteer, start reading.
Limitation of pharmacognosy. Drugs obtained from natural sources have a variety of limitations. The majors are a natural compound may be highly active, but usually this is associated with high toxicity problem. Example, the toxic and the therapeutic dose of digitalis are very close, which creates serious problems to use these medicines by the patient alone. Some compounds are found in nature that give beneficial pharmacological actions, but their potencies are too low to be employed therapeutically. The yield of active natural products may be very low, and thus the production cost of those drugs will be very high. The source of a natural drug may be very limited with respect to geography, season, and climate, etc.
So I think I'll explain you with something which may interest you. And slightly I will divert, not diverting, but I'll give you an example. How many of you know belladonna? Belladonna plant extract, which was added to the eyes. You remember in an older, literature, Greek literature, or if you are hearing the stories of that, to make the eyes very seductive. The pupils will become very dilated and the pupils will become bigger. Now that's supposed to be sedation, I don't know. But it is very well documented to make the eyes more sedative. This was used. This same principle was used to take out the cataract from the eyes. Now I'm telling you at one end one thing and the second end the real crux of how it is medicinally used. Yeah. So this itself was causing a loss of eyesight. So this is a great example of how the plant can be used in one way as a cosmetic. At the second end, the same principle was used for a medicinal purpose, but the toxic dose were very high. So if somebody thinks that I'll become more sedative by putting more drops of it, I think you'll lose the eyes. So this is how, just to make it interesting, I gave you this example. But in you all must be knowing, even in our house, if we eat certain plants very much, too much, then we have problem. So that's what the first para is telling about. But please take a minute to read this, and if you have more questions, please ask me. The second para, I'll explain you. Please, if you have asked question in between, but I'll explain you the second para. That most of the compound which we get it like from the plant. Let's take it as an which I wanted to give you artemisinin as one of the which I'll give you as your assignments. But there are many components, antiviral compounds which we get or antibacterial compound. Ginger, garlic. Garlic has allicins, correct? And which has an anti-allergic as well as antibacterial property. I think you all know about it. Correct? अभी हम लोगों ने बहुत garlic और ginger बहुत खाया है ना अभी during COVID. So, allicin, but it is a potency wise, it is very less, and how much garlic you can eat. You can't eat 500 grams of garlic. In that scenario, the synthetic chemistry comes to your help that that this basic principle is having an ability to act on bacteria and kill them. So then you use your semi-synthetic chemistry approach and the modern chemistry knowledge to make derivatives which are more potent active, that is more bactericidal at a lesser concentration. So that's an importance. Then there is a genomic, because you all are biology student, I need to tell you, there is a big branch of genomics which works to see that selectively that antibacterial component gets very large amount production. Naturally, it's very easy, correct? In your the way you all know now genomics and so many tools are available, we can make it overexpress into the genome, and then we will have a large amount of that antibacterial compound. So a lesser amount of ginger or garlic can give you a much better antibacterial activity. That is what a second para means. The third is yield. Yield is meaning, the milligram, the active component can be very, very effective. Yeah, in belladonna it is very effective. But let me give another example. ठीक है बाद में देता हूँ example. Let me first explain you. The active component may be, when you take 1 kg of leaves and you might get only 1 milligram. Now that's not sustainable, correct? We are talking of economically wide population marketable product. So in that scenario, this is one of the limitation that we get. Sometimes the yields are very low, but every limitation has a solution. So I'm telling you this solution. The yields are very low. Yield means the total milligram per gram of the raw material you use, what you get it. It should be acceptable range. So that is what the yield means. Yeah. And the last one, the source, correct? See, in many places the roots are used as an medicinal value, and that many of our Ayurvedic medicines have roots. Now, what has happened over the years? Like, those plants have been rooted out from the jungle. Like, the main when I was talking about collection of the plant and the knowledge of in which area they are available. So you have to go to that particular forest or particular region And when you go to that particular forest and that particular region, I think what happens is that plant become extinct because everybody goes and root it out, and that's how. So normally it is preferred that leaves or early part of the leaves branch is useful. Did you understood these four different aspects discussed here of the limitation? Yeah, nahi?
Yes, sir.
Yes, sir.
Okay. The this following slides you have to read it and remember. इसमें मैं कुछ समझाने की जरूरत नहीं है. The active constituents, that is morphine. Yeah, in the previous plant, the plant name is given, Pepavir, and pharmacological activity. So important active constituents of the plant used in this medicine. This anti-cancer compound. So here the examples are of various kind, like, right from a sedative to anti-malarial. If you come to the last, pharmacological activity, anti-cancer, yeah, vasodilator. If you don't know vasodilator, don't worry, it's an cardiac related, yeah, or cardiotonic, parasympathetic. This will be now active. So don't worry so much into pharmacology, but overall you should know that anti-cancer you all know, sedatives you all know, anti-malarial you know, correct? Anti-arrhythmic, arrhythmia, okay. Anti-arrhythmic nahi malum hai to koi baat nahi. That's a pharmacological word. That's a heart related again. Parasympathetic is nerve related. So just remember that the cardiovascular, neurological system, then even digestive system, we have a lot of examples in Ayurveda. Or as a anti-malarial and painkiller and anti-cancers can come. So this you will have to by heart. At least, I don't like to say that mug up, but try to remember 3 example at least, because exam mein definitely aayega. You have to give with the example, active constituent, plant and pharmacological activity. If anybody has any problem with this, please let me know. Tell me.
So, was not quinine banned? Is it still in use?
Quinine is not banned. Nah? It is less used now because we have a much better anti-malarial available. But quinine is still in use, very much in use. Huh? In Plasmodium falciparum, we have many medicines, but when it comes to Plasmodium vivax, which is anti-malaria causing, I think still quinine is being used. Huh? So don't. It's a very bitter and it causes vomit, but it's the cheapest available malarial drug. Huh?
Okay.
But yeah, what I'm trying to tell the class is that this is a limitation of our academics, but आप लोगों को ये बाय हार्ट करना होगा. इसमें से कम आपको 3 एग्जाम्पल लिखने ही पड़ते हैं, अदरवाइज आपके मार्क्स गए समझो. सिमिलरली मैंने काफी डिफरेंट एग्जाम्पल्स आप लोगों के लिए, हाँ, CNS एंड एनेस्थेटिक. मुझे ये स्लाइड सेट बहुत अच्छा लगा, सो आई एम शेयरिंग. आई विल शेयर द होल स्लाइड सेट विद यू. The first 4 slides were the real interesting and important from an understanding point of view, definition, then what a little more detail about distribution and different structures and those part of it, then with the example, then how it is connected to modern medicine, then we learned about application and limitation. But these examples, I think there is no other way. Please try to write it down 3-5 times. I think you all know better than me that how to remember. So these are the 3 slides. इसमें कुछ explain करने जैसा नहीं है, but I'll keep the slides on, and if you have any question, please ask me. Huh? Any question, don't worry कि इसके related है या नहीं. Any question you have. क्योंकि अभी next class में हम pharmacognosy touch नहीं करेंगे. I'll send you an email that what is your assignment around pharmacognosy, and that you all will complete in 15 days. 15 days ठीक है assignment करने के लिए कि ज्यादा time चाहिए? मानसवी.
Sir, 15 days is fine.
Fine, ठीक है. तो आज मैं भेज दूंगा 15 September. So I'll give additional whatever 5 days, correct? So 15 September 5 p.m. last shutdown, and all my assignments has to be handwritten, and you have to scan it and send me. Huh? ठीक है? And if you all are coming on 16 September, bring it with you. That will be the first beginning of our class, maybe. Okay.
Yes, sir.
So shall we close the class if you all promise that you will read these 3 slides, or I just keep the slides on and you keep on reading and ask any questions?
Sir, I have a question.
Sure.
Sir, we have studied about a therapeutic and toxic dose in immunology. So, sir, while testing a medicine, how do we get to know? Like, suppose let's take an example of quinine. So, you said like we will test it on an organism and see whether it is lowering down the fever. How we will get to know about its toxic dose?
Excellent, excellent question. In fact, what is your name? I cannot read, but I think good. Your interest in
Shakti.
this field is Shakti. Your interest in this field is very high, and I'm impressed. I think I'll tell you now. क्विनाइन का तो एग्जांपल मैं ले ही लेता हूँ पहले टॉक्सिकोलॉजी एक्टिविटी नाउ आई थिंक यू ऑल हैव अंडरस्टूड फ्रॉम द स्क्रीनिंग पॉइंट ऑफ व्यू एंटीवायरल हो तो कैसे करेंगे amylase ho to that screening models tells you that this compound is active, correct? At the same time, when you test multiple dilution, for example quinine, if I take 1 in 10 if I take a plant producing quinine and then I make a dilution from there in my laboratory, 1 in 10 1 in 10, yeah, so 10-fold dilutions I'll keep on making. So at one stage it will be not active, correct? It will get diluted, the active component will get diluted to so much extent that it will not be active. But the previous dilution tells me that this is minimum active concentration. शक्ति इतना समझ में आया? Minimum active concentration first you have to find out. नहीं?
No, sir.
नहीं. अच्छा चलो मैं एकदम लेमैन का एग्जांपल पे जाता हूँ. आपको मिर्च खाने को दूं मैं, हाँ? तो डायरेक्ट यदि मैं मिर्च का पाउडर आपको खाने को दूं तो इट विल बी वेरी डिफिकल्ट, करेक्ट?
Yes, sir.
But then if I keep on diluting, I'll take one milligram of mirch powder and put it another constituents, say 9 gram. पहले मैंने 10 gram मिर्च आपको खाने के दी थी तो you started screaming, correct? Then if I give you one gram added with another 9 gram sugar and other thing, I think you'll still able to taste the pungentness of mirch. ठीक है, समझ में आया? Now
Yes.
if I take from here this ye 10 gram which you can tolerate, if I put another, I just take 1 gram and put another sugar. So I'm diluting, serially diluting the pungentness of the mirch, and then if I give you to eat, at one stage it will come, you will not be even able to realize कि इसमें मिर्च पाउडर है. समझ में आया? ये तो बिल्कुल ये समझ में आया ना. Now come to एंटी मलेरियल एक्टिविटी. हाँ, यदि मैंने डायरेक्टली प्लांट लिया क्विनिन का, and this is what a screening model, again I'm explaining you. If I take a full plant and if I put it into 1 ml of water and take that 1 ml of water and put it onto the malarial parasite, Plasmodium vivax, actually Plasmodium vivax, if I put it, वो तुरंत मर जाएगा. एक्सेप्टेबल है, क्विनिन से मर जाएगा. हाँ,
Yes, sir.
अब instead of putting this 1 ml directly onto parasite, what I'll do is ये first tube जहां से मैं अभी बात कर रहा हूं उसमें से मैंने 1 ml ले लिया और 10 ml water में डाल दिया. Now it is 1 in 10 dilution. So the drug got diluted. Huh?
Yes.
You understood ना? Drug got diluted. जैसे मिर्च dilute हो गई वैसे drug dilute हो गया.
Yes, sir. So its effect is less.
So effectiveness will reduce. It will take another, instead of instantly killing vivax, it will take around 5-10 minutes to kill, correct? And it will not kill all the parasites. ऐसे ये मैंने फिर से डाइल्यूशन किया तो एक स्टेज ऐसा आ जाएगा कि जैसे कुछ भी असर नहीं होगी वाइवेक्स के ऊपर, जैसे आपको मिर्च की कोई असर ही नहीं हो रही थी, उसी तरह से this quinine will not be able to, because it is got so diluted, it will not be able to kill or inhibit the vivax parasite.
Yes, sir.
एकदम सिंपल तरीके से समझ में आ गया आपको?
Yes, sir.
तो जो डाइल्यूशन मेरा नहीं एक्ट करता है उसके प्रीवियस डाइल्यूशन विच इज एक्टिंग, हाँ, इट इज कॉल्ड एज अ मिनिमम इनहिबिटरी कॉन्सेंट्रेशन. उसमें जो क्विनिन का कॉन्सेंट्रेशन है which was minimum required to inhibit the Vivax parasite? MIC
Yes.
constant. समझ में आ गया ये? तो ये MIC हो, correct? Now this is effective concentration I talked about, and now you have asked a big question of toxicity. So, like, I know that this quinine at certain level it is toxic, correct? So this minimum inhibitory concentration के ऊपर ही यदि मैंने human cells के ऊपर डाल के, and if it's the cells start lysing, then it is of no use. That will be not drug. So, there has to be 10-fold difference between minimum inhibitory concentration and minimum tolerated concentration by host. So that is how we find out the toxic versus active dose. And there has to be at least, that's a big science, toxic versus active dose. But just to give you an idea that we put it into animals, we put it into human tissues and see that it is not really killing the tissues or human cells or blood. Actually, if we start with the blood cells, एकदम ब्लड के ऊपर लाइसिस होना, जो मिनिमम इनहिबिटरी कॉन्सेंट्रेशन अभी हमने फाइंड आउट किया, यदि उसी को मैं ब्लड के ऊपर डालूं एंड इफ द ब्लड सेल्स स्टार्ट लाइसिंग, दैट इज ब्रेकिंग डाउन, then it is of no medicinal value. But if I go upside the original extract which I have taken, and if I put it onto blood cell, it might lyse. But the 1 in 10 dilution is not lysing. So the difference between the blood lysing concentration versus the minimum active concentration, if there is a 10-fold difference, that is called as active versus toxic dose. थोड़ा कॉम्प्लिकेटेड है. यदि जब आप कॉलेज में आओ तो मेरे पास बैठोगे तो मैं आपको पेपर पे ड्रॉ करके सिखा दूंगा तो आपको आएगा. बट राइट नाउ दिस इज इनफ. ठीक है?
Yes, sir. I understood.
Okay, thank you.
Thank you.
Any other questions?
Sir, actually our next class started.
Oh, sorry. Please log out.
Thank you so much, sir.
Thanks for telling. I'm sorry.
Thank you so much, sir.